Okay, welcome to my student. So I am continuing the lecture. This is the second lecture on tyrosine kinase. So today we will learn some specific case. Okay, so let me take one. Okay, so there are common, we already know there are three common structural changes may happen in regulatory chromosomes. One is translocation, inversion and deletion. So today for a specific case we will understand the translocation. So this is one chromosome, this is another chromosome. If one portion of the chromosome is transferred and sit on the another chromosome, it's called translocation. Now you see this part, say this is 1, 2, 3, 4. So one is separated out from this chromosome number 1 and it is sit on the chromosome number 2. So this is 1 and this part 4 and vice versa. So this is 3 and this is 2. This is called the translocation of chromosome. Okay. Now we will understand a particular translocation. Okay, why we are learning this particular translocation? Because this will activate some intracellular signaling molecule, signaling protein. Okay, so this is the chromosome number 9. Okay, so there is a gene called ABL and this is the chromosome number 22. There is a gene called BCR. Now what is happening, this part of chromosome number 9 going, it is transferring, it is separated out and sitting on this region. You see, it is forming this. So this part is as it is and this part is come and sit here. Okay, so and remaining this part is here and this part is here. So this translocation T922, okay, it's called Philadelphia chromosome. It's very important. Why? I will tell you. This Philadelphia chromosome T922, basically it's a coding for a hybrid protein. Basically it's a tyrosine kinase signaling protein which is always on. Okay, so if this kind of translocation happened and Philadelphia chromosome is formed by translocation, then the signaling, the tyrosine kinase signaling protein is always on. Now we will go to our previous few slides. See what is happening. If you recall the early the first lecture, the tyrosine kinase available in human cell cytoplasm as in monomer state, which is inactive. But if now the signaling molecule come and sit on the binding site, then it will convert to dimer. Once it is a dimer is formed, now it is not inactive, now it's become active. This will interact with ATP and it will take the phosphate group from the ATP. Eventually if the one phosphate is left from ATP, it will convert ADP, and now this dimer will get phosphorylated, which will activate, which will further chemically react with some inactive relay protein, maybe some cancer protein. Okay, it will come and sit on the phosphate binding region of the dimer, and in turn it will activate different kind of cellular response. It will activate different transcription regulation unit, which may interrupt the conversion of DNA to RNA. Okay, this may cause some abnormal and rapid cell division. Okay, so now by this kind of translocation, if that Philadelphia chromosome is formed, T922, so it's basically a tyrosine kinase signaling protein which is always on, and now what is happening, it will interrupt the transcription regulation unit, which will abnormally interrupt, which is abnormally which is abnormally this abnormal cell division will take place. Okay, so the cell division will take place uncontrollably and abnormally, which may cause the chronic myeloid leukemia, CML. Understand? Now, how to stop it? How to stop it? So there are two ways left out. One is to stop this translocation. We have to stop this translocation, or we have to reverse back the translocated chromosome to its original structure so that the chromosome can do its regular action, or we have to stop the phosphorylation. That means we have to stop, so that we have to do something. We have to apply some drug so that the ATP cannot donate the phosphate group to that activated dimer. If the activated dimer is not able to get phosphorylated, then it cannot activate the relay protein, further that cancer protein, and then that it cannot enter inside the nucleus, and that it cannot interrupt the DNA to RNA conversion. This is the two way. But you know, to handle with gene, it's very difficult. So far it is not possible. A lot of research are going on. Okay, so only the way left out to stop the phosphorylation. So there is some drug. If you apply, then it will stop the phosphorylation, and if the phosphorylation is stopped, then the unregulated and uncontrolled cell division also can be stopped till some extent. This kind of drug, which can stop the phosphorylation of active dimers, it's called tyrosine kinase inhibitors. Those drug name will be ending with NIB, and those are oral drugs like imatinib, nilotinib, dasatinib, all these things it will do that. Okay, so this is the very basic of tyrosine kinase inhibitors. So this for details, for the details you can follow this book. I have followed this book because I always follow this book. This is the book Kumar and Clark's. This book Kumar and Clark's, page number I think 464. Page number 464 will be there. where the intracellular signal inhibitors are detailed. Let me open. Yeah, in page number 464, this is the edition. Which edition? I don't know. It is the 2009 edition. So page number 464, there will be intracellular signal inhibitors. There is a tabular form. There we will get all the details, and we will learn these things slowly in the subsequent lecture. So thank you so much. Hope you understood the tyrosine kinase inhibitors. Thank you so much, students. Thanks for watching. Thanks for your patience.